Okay, Dickie, we're taking about a simple overview of DNA replication. I'm going to get really deep into it, but in later videos. But this is just the general idea and kind of how the structure of DNA lends itself to how it is replicated. So we have a double helix, and the double helix is going to look like this. So I draw a wavy line, draw another wavy line, right? That kind of looks like it, and then you draw the lines in between the two rungs, and you get your double helix. But if we untwist it, then we kind of have a ladder looking shape, and I'm going to draw the bases in between it. Cool. So this is kind of what DNA looks like. It's the double helix, but this would be twisted. And let's say, remember its structure, we have the phosphate sugar background on the outside, but then the bases, they bond A, T, C, and G. So we know that C and G and A and T go together. So if I have the simple sequence of A, T, C, and G, what's the other side? The other side is going to be T, A, G, and C. Cool. So now we have our strand here of DNA. Now we'd have our sugar and phosphate backbone on the outside. But what if we want to replicate this? So what if we cut it in half and move one strand over here and one strand over here? So now we have T, A, G, and C over here, and then we have A, T, C, and G over here. We're just going to line up the other copies. So that becomes T, that becomes A, that becomes G, that becomes C. Same thing for the other side, A, T, C, G. And now look, now we have two identical copies of the first set of DNA. And so this is the general idea that happens in DNA replication. I hope that was helpful.